Hello, everyone. It's our pleasure today to introduce Dr. Martina Dal Bello. She's assistant professor of ecology and evolutionary biology at Yale University. She specializes in microbial community ecology. Martina's research delves into how bacteria respond to and modify their environments, influencing species interaction and the structure of microbial communities. By integrating laboratory experiments, natural community data, and theoretical frameworks from community ecology, microbial physiology, and system biology, she aims to build an ecological toolbox for managing and manipulating microbiomes. Martina earned her PhD from the University of Pisa and continued her research as a postdoctoral fellow in Jeff Gore's group at MIT, focusing on effects of temperature and nutrient availability on microbial diversity and community structure. Please welcome with me, Dr. Martina, and this virtual stage is yours.
Thank you. Thank you very much for the introduction. And it's a pleasure to be here and tell you something about my research. So this is something that I did during my postdoc. And so this work, but this is an area of research that I'm really excited about and will continue as I started my group at Yale. And so this talk is going to be about the distribution of fast and slow growers and how, let's say, this changes predictably with the seawater temperature and salinity. Okay, first, I want to start by telling you why I am really excited about the microbes and especially microbes in the ocean. So in general, across ecosystems, and probably I shouldn't tell you to this audience, so you already know that microbes degrade organic matter, both in terrestrial and aquatic ecosystems, recycling nutrients and regulating the long-term storage of carbon. But especially in the ocean, microbes are responsible for producing the oxygen that we breathe. And in general, across ecosystems, they regulate the production of important greenhouse gases like carbon dioxide, methane, and nitrous oxide. In general, we still don't know how different environmental processes affect how communities assemble and how community function, not just across the oceans, but across several ecosystems. And one important thing is that thanks to the advent of sequencing techniques from metagenomics to 16S amplicon sequencing, we've starting accumulating a lot of data about microbial communities and especially marine microbial communities. For example, I don't know if you know this famous boat, but this is Tara Ocean, the Tara Ocean expedition. This expedition have been sampling the ocean microbiome across latitudes and depths and generating interesting patterns. For example, here on the right, you see how the diversity of heterotrophic and phototrophic bacteria changes, and archaea also changes as a function of latitude. And you can see that in this pattern, that the diversity tends to peak at the equator and the tropics. But we also have like buoys, like this one in the middle of the Baltic Sea, that collect environmental data, for example, temperature and the concentration of nutrients. But also at the level of this buoy, scientists can sample marine communities and track the abundance, how the abundance change in time. And for example, this is a result of one of the studies showing how the composition of marine communities changes predictably with seasons. What I think is what we see is that we are finding many patterns, but we're struggling to find unifying principles explaining these patterns. what I'm going to argue across this talk is that there is an interesting concept in ecology, that is the concept of fast versus slow growth, that can be used as a unifying pattern, as a unifying concept to try to explain some of the patterns that we see across marine microbiomes. So this concept has been extensively used in ecology, especially in the context of recovery from disturbance. For example, in a plant community, when disturbance wipes out this community, the first species that are going to colonize this empty space are usually faster growing grasses that only later will be replaced by slower growing bushes and trees. And this concept of fast and slow growth is also pervasive in microbial ecology. So, for example, we distinguish between copiotrophic microbes that are usually fast growers and tend to thrive when nutrients are abundant, and slower growing oligotrophs that instead are usually found where nutrients, or at least are more abundant when nutrients are scarce. So, today, in this talk, I'm going to use this concept of fast and slow grower, and specifically the distribution of fast and slow grower, to show that to show some interesting patterns. So first, that slow growers are more abundant where and when temperatures are higher in the ocean. Then I will also show you that faster growing bacteria are usually favored by increased salinity in marine microcosms. And finally, throughout this talk, I will show you how Lotka-Volterra models can help us predict community structure as the environment changes. So, about the first story of today, it's about temperature. So temperature is a cardinal variable for microbial life. For example, it regulates enzyme kinetics and bacterial growth. And actually, bacterial growth and enzyme kinetics can be described by a thermal performance curve that you can see depicted in this plot. So growth increases with temperature up to an optimum, and then starts decreasing because of the denaturation of proteins up to a temperature maxima at which bacteria stop growing. And so it has strong effects on physiology, and the strong effect of physiology actually helps us understand, helps us distinguish microbes with respect to their preferences for temperature. So in this cartoon, I'm showing you how microbes can be distinguished based on their favorite, their temperature range. So we can go from psychrophilic microbes that usually found at lower temperatures up to hyperthermophilic microbes that instead thrive at incredibly high temperatures. So given that temperature is such an important variable for microbes, one question is whether these effects on physiology can affect how species interact when temperature changes. So an experiment from my postdoc lab that were conducted by my colleagues Claire Abreu and Simon Lux showed that increasing temperature can favor slower growing taxa in pairwise competitions. And specifically, Claire and Simon competed two species. In this case, they are depicted as this red bug that is and a blue bug. At 3 different temperatures, and specifically 16 degrees Celsius, 25 degrees Celsius, and 30 degrees Celsius. What they found is that across all these temperatures, the species that here is the red bug was actually faster grower, and the blue bug was instead a slow grower. And but the important finding is that they found that the red bug was actually constantly winning the competition at the lowest temperature at 16 degrees. They found that the two bugs coexisted at 25 degrees, and finally that the slower growing species was actually winning at the highest temperature. So basically what they found is that across these different temperatures, the slower growing species was actually favored. And this was not just an isolated instance. They checked this result in several pairwise competition experiments with many species pairs. And what they found that this result was consistent. So temperature was consistently favors the slower growing species. But to me, this result actually generated a very interesting question, that is, can you actually see this pattern in natural communities? So during the pandemic, I partnered with Clara Abreu that was involved in these experiments, and we decided to see whether we could actually see this pattern in natural communities, collecting data from several public repositories. And specifically, we had a very clear hypothesis that we could test. And if we think about marine communities where temperature changes across several axes, several environmental axes, we could actually test whether slower growing taxa were more abundant in the summer, at the equator, and at the surface of the ocean. So with this clear hypothesis in mind, Claire and I started collecting datasets where marine communities have been sampled across seasons, latitudes, and depth. We collected a total of 7 datasets, but for the purpose of this talk, I'm going to just show you the results from 3 of them. Specifically, one of these datasets is this time series collected in the middle of the Baltic Sea at the Linnaeus Marine Observatory. This is actually the buoy that I showed you in the picture before. This is an amazing time series. So scientists have been measuring environmental data, including temperature, and sampling microbial communities for more than 11 years now at a frequency of about 2 weeks or monthly. And so in this case, we had communities sampled along a gradient of temperatures, so across different seasons. Then we got access to this amazing transect that crosses the Atlantic, basically from the coast of Patagonia to the coast of the UK. And in this case, the marine communities were sampled also across 4 different depths at about 200, below 200 meters of depth. And finally, we've also seen this before. This is the map of the Tara Ocean. So, and you can see that samples are distributed across different latitudes, but the most interesting thing about Tara Ocean is that samples are collected across many different depths, from the surface to 1,000 meters below the sea level. So with this data set, we really had access to 3 main axes of temperature variation, so across time, across seasons, and across depths. But there is one problem. So I told you that I wanted to focus on fast and slow growers bacteria, but we cannot go in the lab and measure growth rates. So we needed to find another proxy for computing the growth rate of bacteria from natural communities. And lucky for us, a few years ago, a paper that came out in Nature Microbiology by Ben Roller and colleagues showed that the maximum growth rate of bacteria is proportional to the number of copies of a 16S of gene operon. So basically what this relationship is telling us is that if we have a species that has a high copy number compared to a species that has a low copy number, this species with the highest copy number is potentially growing a faster grower compared to the other species. This also means that if we have a community, we can calculate the mean of the copy number weighted by the abundance and of all the different species present in a community. And this single number basically renders the abundance distribution fast and slow growers in that specific community. And basically what this is telling us is that if a community has a high mean copy number, is composed of more fast growers compared to a community that has a lower mean copy number. And this particular metric has been shown to respond to changes in the environment, particularly in nutrients. So with this tool in our hands and all the data that we collected, we went on and calculated the mean copy number for each community and basically paired it with the value of temperature measured at that in correspondence to this particular sample, And these are the results. Here, in this plot, I'm showing you how temperature varies across these 3 different datasets, and there is nothing really surprising about it. So for the LMO station, so the Baltic data, we have temperature sampled across several different years, and not very surprisingly, we have the highest temperature in summer and the lowest temperature in winter, since we are in the boreal hemisphere. Then this is how temperature varies across the Atlantic dataset. You can see that, again, everything is kind of what we might expect. Temperature is higher at the level of the tropics and the equator, and is lower as we approach the poles. And finally, this is data from Tara Ocean, and specifically a slice of data between 0 and 40 degrees south. That had the most dense samples across the water column. And in this case, temperature varies predictably with depth, basically decreasing as we go down the water column. Now, what I'm going to plot are how the mean copy number changes for each sampling station. And in general, the mean copy number follows pretty well the variation in temperature. And specifically, you can see that the lowest values of the copy number tend to correspond to the highest values of temperature. And indeed, we found a strong negative relationship between the mean copy number and temperature. This negative relationship is basically telling us that as temperature increases, slower growing bacteria are becoming more abundant. Excuse me. So the other thing I want to draw your attention on is the slope of the relationship. So even though these communities come from very different basins, you can see that the slope of the relationship is really similar, which I think is an interesting finding, considering that we are comparing the Atlantic Ocean, which is a very open basin, with, for example, the Baltic Sea, that is a more brackish environment with a lot of nutrient inputs. But you can also tell me that, well, temperature is not the only environmental variable that is changing across these different environmental gradients. And here I'm again taking advantage of the amazing data set that comes with that is collected at the LMO station. And you can see that, to corroborate what I just said, is that other environmental variables show strong trends with season. For example, you can see that the dissolved organic carbon peaks, in this case, during the summer months, while nitrates and phosphate actually are more abundant, are more concentrated during the winter months. So you might ask, well, maybe it's not temperature that is driving the distribution of fast and slow growers, but there are other environmental variables that might be important. So to try to check for this effect, we ran some generalized additive models that allowed us to account for the effect of other environmental variables on the distribution of fast and slow growers. And in this particular plot, I'm showing you the results of this analysis. Each tile represents a value of the parametric coefficient estimated from the generalized additive models, multiplied by the standard deviation of the predictor, so that we can compare them. And warm color indicate positive values, and cold colors indicate negative values, and the asterisks indicate whether the coefficient is statistically significantly different from 0. So you can see from this analysis that the effect of temperature is consistently negative and statistically significant where other environmental variables are taken into account. And even when other environmental variables, for example, ammonium in the case of the LMO data, or phosphate in the case of Tara Ocean, have important effects on the distribution of fast and slow growers. And to make sure, we also checked other data sets where you have other variables that are extremely important in determining the distribution of fast and slow grower. Specifically, this SOLA data set is a time series from the Mediterranean Sea, and SPOT is another time series, is actually, sorry, a transect in the Pacific Ocean, if I'm correct. And in this case, you can see that, for example, in the case of SOLA, the day length, so the number of hours with light is an important variable affecting the distribution of fast and slow grower, but the temperature is still relevant in consistently favoring slower growing bacteria. So the last thing that I want to tell you briefly about this story is that it seems that the effect of temperature on the distribution of fast and slow grower is actually quite general. So here I'm showing you two maps showing the distribution of Prochlorococcus and Synechococcus, that are two of the most abundant cyanobacteria in the global ocean. And you can see that their distribution is well known. Prochlorococcus is more abundant in warmer waters, and specifically around the tropics and the equator, and Synechococcus extends its abundance also towards more temperate regions. And actually from estimates in the lab, Prochlorococcus is actually a slower grower compared to Synechococcus. So the pattern that we found that temperature favors slow growers is consistent with the distribution of Prochlorococcus and Synechococcus. The other thing that I want to show you is that this applies also to different systems. So, for example, these are the results of an experiment conducted in the Harvard forest, where plots of soil have been warmed for more than 20 years, and the mean copy number, the mean RNA copy number has been measured, and you can see that the warm plots compared to heated plots compared to control plots show a reduced mean copy number, indicating that temperature contributed to a reduction, to a general effect on favoring slower growing bacteria. So if you haven't stopped me, and maybe you are asking, what you should be asking yourself is now why temperature is actually favoring slower growers. So to try to go about this question, we used a modeling approach. So here I'm showing you the classic, let's say, model, Lotka-Volterra model, where we have the per capita growth rate as a function of the maximum growth rate, a self-inhibition term, and the inhibition by a different species. In this case, for example, I'm looking at the per capita growth rate of the slow grower, and here you have the inhibition of the fast grower. We also introduced in this model a mortality. Originally in these models, the mortality was mimicking the effect of these daily dilutions that we perform in the lab, but it's actually relevant also in the context of the global ocean, where microbes are constantly under predation from grazers, from phages, and also there is senescence and, let's say, dilution from through currents. We can write, let's say, bring this equation back to its original form by absorbing this mortality term. And this operation, what basically allows you to show is that the outcome of the competition is only determined by the value of the inhibition coefficient, so the competition coefficient, and that actually the value of the competition coefficient depends on the ratio between mortality and growth rate. So everything is determined by changes in growth rate and mortality. So actually we can plot this. In this kind of diagram, I'm plotting the logarithm of the inhibition of the slow versus fast on the x-axis, and the logarithm of the coefficient of inhibition of the fast by the slow grower on the y-axis. And I told you that in the Lotka-Volterra model, the outcome of the competition depends solely on the values of the competition coefficients. And we can show that when both coefficients are negative, two species coexist. When both are positive, you have bistability. So the outcome of the competition depends on the initial condition. And then you can have either the fast grower winning or the slow grower winning, depending on which inhibition coefficient is actually positive. Now, we have this very nice framework, and we know that the competition basically depends on mortality and growth rate. In our case, mortality is fixed. In the experiment, it's just the dilution, and in the ocean, we can kind of assume that is kind of a process that affects equally everyone for simplicity. So what we can actually change is the growth rate. And growth rate, we know that can change with temperature. So we model this dependency of growth rate with temperature using this Arrhenius model. And the Arrhenius model shows that the temperature depends on an activation energy, the Boltzmann constant, and the temperature in Kelvin. And we can also account for differences in the growth rate between fast and slow growers by multiplying this exponential relationship by the copies of the 16S operon. And if we are comparing a fast and a slow grower, we put in some numbers in this equation, we can see that as we expect, temperature increases growth rates up to an optimum. Here we are considering only the temperature that allows a positive increase in growth rate. And as expected, and as we see in the experiments, if we model growth rate as a function of temperature with Arrhenius, we see that the faster grower is consistently growing faster than the slower grower. And then we have this mortality term here that indicates how much mortality there is in the system. Well, if we go back to our kind of previous slide where we have the dependence on growth rate and mortality, we can see that because of how this ratio is built, we can, if we increase the growth rate, for example, by allowing increasing temperatures, we can go from a condition in which the faster grower wins to a condition in which a slower grower wins. And this is just by increasing growth rates and keeping mortality fixed. So basically, the reason why temperature is actually favoring slower growers, it's because it's increasing the inhibition of the slow grower and decreasing the inhibition by the fast growers. Okay, this was probably quite fast, but the math is there, and believe me, you can prove that this is the case with an analytical solution. The other thing is that when we have multiple species, we can generalize the Lotka-Volterra model to include, let's say, hundreds of species. We can still have growth rate as a function of temperature, but this time, as I said, we have a group of species, and we can distinguish among them using the copy number, and we can use a distribution, specifically a geometric distribution, to fish the copy numbers to assign to each of these species. And this P of the geometric distribution basically is telling you the fraction of species that have copy number 1. So it has a biological meaning. So if we run simulations with this more complicated Lotka-Volterra model, so we let the communities equilibrate in the simulations, and we calculate the copy number at the end of the simulations, we find that actually as we increase temperature, the mean copy number actually decreases. So even this form of the generalized Lotka-Volterra model is consistent with the with increasing temperature favoring slow growers. The last thing that I think is quite interesting is that we can use the Lotka-Volterra model to fit the data, and we find basically that we only need one parameter to fit the data in a very reliable way. So this parameter is actually the P of the geometric distribution that I told you before. So once we fix the P, that is basically telling you for each data set the fraction of species that has copy number 1, the Lotka-Volterra model fit is actually following the data very well. Here you can see in each of these plots, the dots are the observed mean copy number, and the black line instead represents the model fit. And I want to draw your attention to the, let's say, overlap between the observed slopes, the one that I showed you before, so that describe the relationship between temperature and mean copy number, and the fitted slopes. So they're really, really similar. So it means that they're really doing so this simple model is really doing a good job in predicting variations in mean copy number due to temperature. And this is just to show you that, again, the only parameter that we need is this P of the geometric distribution. And thanks to this, this model actually recapitulates pretty well the relative abundance of each copy number that we observe in the dataset. Again, the colored bars are the real data, and the gray bars are the fitted data, showing you the distribution of the different copy number across the datasets at the end of the simulations. Okay, if there are any questions about this first part, I still have 10 minutes, so I'm gonna briefly talk about the next part of the talk. So after we've seen how good we were doing with temperature, we said, okay, can we try to understand how another parameter, another environmental variable changes the distribution of fast and slow grower? And with Jana Husman, who is currently a postdoc in the Gore lab, we started looking at salinity. Why salinity? Well, a few years ago, a paper showed that the salinity, surprisingly, is actually a major determinant of community phylogenetic diversity. So in this paper, Lozupone Knight took data from very different environments, from marine sediments, marine waters, but also fresh water, and then hot springs, acidic pools. And they looked at the phylogenetic diversity and did some clustering. What they found is actually that the major axis of separation between all these different samples was actually whether were saline or non-saline environments. So really salinity, more actually than temperature and pH, determines the diversity, the phylogenetic diversity of bacterial communities. The other important thing for us is that salinity affects growth rate. So it has a strong effect on growth rate. So here I'm showing you these salinity performance curves. These are the equivalent of the thermal performance curves, but on the y-axis, on the x-axis, we have different concentration of salts, and on the y-axis growth rate. And you can see that, and these are different marine species. So this is data generated from Jana, that shows that past an optimum, higher salinity is usually coupled with lower growth rates. Now, if we go back to our modeling framework, let's say that we are in a situation where there's lower growing winds. So, for example, we are at high temperatures, low growers are really thriving. Is there a way that we can favor fast-growing species? So, in principle, salinity decreases growth rate, and so we can imagine that decreases in growth rate promoted by salinity can shift the outcome of the competition from the slow grower winning to fast grower winning. So again, we have an hypothesis that we can try to test, and this hypothesis is actually that increasing salinity should favor faster growing species. So for this specific, let's say, question, we decided to take a different approach. Instead of looking, so we're also looking into like data sets, but also we decided to do experiments with marine bacterial microcosms. So Jan and I took a trip around Boston, and we sampled water from different places around MIT. We took a sample close on the Charles River, so in a kind of freshwater environment. Then we took a trip and we went close to the ICA, so the Contemporary Art Museum in Boston, where basically it's where the river meets the sea. So we are in a brackish environment, and we took a sample there. And finally, we took a trip to Nahant, that is that fully ocean, and we also collected a sample over there. And so we have a range of salinities going from fresh water to marine environments. And we concentrated each of these samples in order to obtain a, let's say, a bacterial suspension. And we use this bacterial suspension to inoculate microcosms that have different degrees of salinity, and specifically 16, 31, and 46 grams of salts per liter. And with salts, I mean what usually we mean with marine salts, so not just sodium hydroxide, sodium chloride, but also other type of salts that usually distinguish marine environments from freshwater. And we took a daily dilution approach, so we grew these communities under daily dilution. And finally, after these communities reach equilibration, we extracted the DNA and performed 16S sequencing. And from the 16S sequencing, we were able to match the 16S copy number, and again get the metric, so the mean copy number that allows us to look at the distribution of fast and slow growers in each of these communities. So, and these are the results of this experiment, which are consistent with our predictions. So as we increase salinity, the mean copy number of the community shows a slight increase, which means that going from 16 grams of salt to 46, we generally favor faster growing bacteria. But this approach gives us also another type of advantage, because at the end of the, let's say, equilibration period, we can also actually plate the communities and pick species. So we can select different species in isolation, and we can grow these different species at different level of salinities to actually probe, to actually measure the growth rates of the species that we kind of enrich for in our communities. And when we do this operation, what we find is actually that even in this case, even if we kind of measure the community growth rate in this more experimentally savvy way, we can see that faster growers are generally favored. So the mean growth rate of community members increases with increasing salinities. Okay, so what I would like you to remember from this talk. First, that I think in general, we can say that slower growing species tend to thrive when the environment is more benign. So when temperatures are warmer and when there is not a lot of salt. The other thing is that I think is these results show that the effect of temperature and salinity on physiology scale up to determine community structure, but to actually understand how these effects scale up, we need to understand how species interactions are modulated by changes in environmental variables. And finally, even though the Lotka-Volterra model is an extremely simple model with a lot of assumptions and is actually a phenomenological model, it's really helpful in predicting community structure as the environment changes. Okay, and with this, I would like you to thank all the people that made this work possible, my postdoc advisor, Jeff Core, and my collaborators for the temperature paper, Claire, Karina, and Yaron. Claire has recently started as assistant professor at NYU, and also Karina started in Gothenburg as assistant professor. So after this paper, we kind of all found a job, which was really great. And the other collaborator on the salinity paper, that is Jana Husman, who is still a postdoc with Jeff Core. And finally, this work was funded by the Simons Foundation. And also I want to thank you for inviting me and for listening to this talk. And maybe if I have 3 seconds, just to say, I am recruiting. So if you are let's say, college student that is looking for a post-bachelor experience as a research assistant, or you are thinking about joining Yale as a PhD student, or you want to join as a postdoc, my lab is recruiting. And finally, I'm organizing together with a few people a Kavli KTP program in Santa Barbara. and about coarse-graining microbial communities going from genes to physiology to communities. And this is going to be in 2026, but registrations are already open because there are not too many spots. So with this, thank again, and I'm happy to take any question.
Thank you very much, Martina, for this great talk. Yeah, with Marika, we'll take some questions. I will start just with a quick one. Do you have any also data that correlates with temperature and cell size of this fast and slow growing?
Not personally, but so there are a few. So in general, cell size correlates with growth rate, but I would say there are a few papers showing that the correlation is there, but I don't know, in the context of trying to look for proxies for growth rate, the best proxies are this copy number and the codon usage bias. So cell size is correlated, but the correlation is not as strong as these other proxies.
Okay. Marco?
Hi, Martina. Thanks a
Hello.
lot for the talk. It was very nice. So I actually had a few questions, but I'll start with the one that is bugging me a little bit more. Nice about your Lotka-Volterra model. So if I understand well, you are in a situation with the in a positioning parameter space where you never have oscillations, right?
No.
Which is usually the reason why the Lotka-Volterra model was introduced was because of oscillations. Which means, if I understand correctly, that you're in a situation where you always have at least one of the two parameters that determine the crosstalk between the populations, which is low, right? So you either have alpha SF or alpha FS, which is low, right?
Not low. They are similar usually. So here basically we're saying that we are in first we are not in the context of predator prey, so where you usually see oscillations, but is in the context of competitors, so species competing, so the same trophic level.
Mm-hmm.
And the other thing is that we are standardizing by the carrying capacity, and we are kind of saying we are not taking into account the self. So there is not an alpha for self inhibition. So it's always set to 1.
Mm-hmm.
But in this case, we are in the position in which you can always get an analytical solution and you don't have oscillations. So there are other conditions in which you get oscillations, though.
Of course. But you are in like the second or the fourth quadrant, right? So which means that one of the two alphas, if I understand well, is your graph here is always.
Let me go back. I'm trying to understand how to go.
No, but my point is that I didn't really catch the gist of why the change in temperature is moving the balance from one side to the other. So maybe because I guess it was too fast for me. You have this Arrhenius law, which I guess is just a phenomenological law to get some sort of sigmoid in the growth. With a slope, however, with a prefactor that depends on the copy number, right? So if you have a higher copy number, then the alpha grows faster. I guess this is what I gathered from the talk. And
try to go back to the slide while let me see.
And I wanted to see if you can spend a little bit more time in discussing what happens to the parameters as they are shifted from one side to the other.
Yes.
The mechanism that makes them shift from one side to the other.
Let me see if I can, because I don't see my slides anymore.
We do, by the way.
Can you see the slides?
Yeah.
Okay, good. So am I back to the?
You're back to the
Lotka-Volterra one?
Yeah, exactly.
Great, fantastic. So I managed to do that. So actually, so when you go from, let me see, where are we? So here you see that basically, do you see my cursor?
Mm-hmm.
Okay, or not?
No, maybe not.
Okay, because I don't.
You can stop sharing and share again.
Yeah,
Maybe
probably
it will
I'll try to
solve
do
it.
that
No,
again.
yeah, wait, we see it. We see
No,
it.
I'll be back. No worries. Okay.
Perfect.
Perfect. Great. Sorry. So here we're just writing down the model with this added mortality term, and you can absorb it basically and in the equations, and then what you basically find is that in this absorption you will write this alpha coefficient that is mortality included as this alpha, and now is SF, and there is this ratio between basically what we call the mortality burden, so delta divided by the growth rate. So the shift from one quadrant to another is basically achieved just by changing mortality or growth rate. So in this case, if we keep the mortality fixed, temperature is affecting both growth rates in a way that is described by Arrhenius. And so this growth rate is increasing faster than this one,
yeah.
but the mortality burden in this one is decreasing faster than this other one. And in this kind of ratio determines the shift from let's say if you are in the slower growing winning quadrant, so changing growth rate is going to move you through a coexistent phase, and afterwards through the opposite phase, so the fast grower wins. So the changes in the shift is only related to how the alphas are changing depending on the growth rates. The things I think that is easier to see probably is that if you keep the growth rate fixed and you change mortality, you are doing basically the same thing. So if you are in a condition where the slow growing wins, increasing mortality is actually favoring the fast growers.
Okay, fine.
We have the whole math.
No, fine. Yeah, it's cool. I think, but it's okay. I'll read your science advanced paper, and
but yeah, so it is basically, it
I want
depends
to hog the
all on how this ratio is constructed, and depends what in the moment you add the mortality to your model, that's what happens, kind of.
Yeah.
Of course, you are considering only temperatures in which growth rate is increasing, because the moment you are shifting to the portion of the curve where growth is decreasing, our prediction failed. Mm-hmm. It's not working anymore. So this depends, this is really fixed to the temperature in which bacteria are actually are below their optimum. Okay. And for the ocean, it's kind of, I think it's reasonable because there are not let's say temperatures are there are not super strong variations in temperature. You can assume that for a lot of microbes, you are in this sweet spot where temperature are always below the optimum. Mm-hmm.
Okay, thanks.
Thank you for the question. Usually it takes a bit more to explain this than the 2 minutes during the talk.
Okay, more questions? Feel free to raise your hand. I'm just jumping in because in the community we all care a little bit about the shape and sizes, and I can still I don't know why it triggers me question why you represent those as a rod motile, the fast grower, and the ones as just a cocoid small, just from I just wonder if it.
Okay, it's in my head it was a cartoon. In general, slow growers are smaller.
Okay.
So that's why I represented the slow grower smaller than the fast grower. And it's true that if you think about copiotrophs, one of the traits is usually so it's more likely that faster growing cells are actually motile.
Yeah.
So there is in my cartoonish rendering, there are some ingrained ideas about how you distinguish between copiotrophs and oligotrophs.
Yes. Yeah, I guess there is a way to figure out if the community, if they are motile, non-motile, which eventually if they are elongated or not by looking at the gene composition. So I have one little question as well. Did you, when you sampled around Charles River and MIT and all this area, have you considered the eutrophisation? Because these waters are high in nitrogen and other nutrients, to distinguish it from the effect of salinity or?
Yeah, so when we did the sampling, we measured the nitrites and nitrates, and the Charles was a bit higher than the rest. But to kind of avoid these confounding effects that you get from nutrients, when we were passaging the communities, we use the same medium, which is a marine broth, where we either increase the salt concentration, or so if to, let's say, have less salt, we dilute the marine broth, but then we add back the nutrients. And when we want to concentrate in salts, we keep the marine broth fixed and then we add more salts. So in principle, by doing these experiments in these kind of very controlled microcosms, we eliminate the differences due to nutrients that were present at the beginning. So yes, at the beginning, communities might have been different, not just because the salinity of the environment is different, but also because the Charles has, in general, more things. And I'm not only saying nutrients, I don't know whether they're pollutants or blah, blah, blah. But then once we are in the microcosm system, we kind of control for temperature, for the nutrients and this type of things.
Okay, thank you, Marco.
It's a very good question.
Marco, you can go ahead.
Again, since seems to be, I have some time to ask you a bit more questions. So it was very interesting when you showed the table that looked at potential other causes for the difference between the slow and fast growth prevalence instead of temperature, and you looked at duration of the day, which was one of the things that I also was thinking about. My question is duration of the day linked to irradiance, which was actually maybe something more that could be more impactful for microorganisms, like in terms of UV irradiance, for example, that you could also see the difference, right, in going in depth, right? Very clear difference in UV not reaching lower portions of the sea. And is that something that should be checked separately, or is something that in a sense goes hand in hand with the length of the day?
So for each dataset, so what you say is generally true. So the day length has to do with the amount of hours that the communities are exposed to light. And I think it's, in this case, in the Mediterranean dataset, the communities are pretty, I think where they collect samples is pretty shallow. And so that's why they look at the length of the day as a proxy for how much light they get. Also because in these communities, we're not excluding phototrophs. So these communities include cyanobacteria and the rest. So that's one reason why it might matter to include the length of the day, because it gives an idea of how much light they're getting. And the other thing, so we should consider it in the sense that if we are also including phototrophic bacteria, light might be relevant. The other thing is that, unfortunately, these are publicly available datasets and not all the environmental variables are available. So with the LMO, we actually had, so we were looking at, this is the most extensive dataset that we have because we partnered with the people that collected them. For example, for the transect, we couldn't access any data. And the only thing that we could check was the effect of latitude. So you can account in the model for latitude as that can have nonlinear effects on your mean copy number. That's how we deal with that. But so in general, this table is not all filled because it depended a lot on what we had available. Related to this, we can repeat this analysis excluding phototrophs or, for example, excluding species that have copy number 1 because they're actually very, very abundant. And we can show that this pattern of temperature favoring slow grower is actually stronger, for example, when you exclude copy number 1. So this one criticism that we got was that we were tracking the abundance of slow growers, but actually when we exclude them, this pattern is very strong. So we kind of avoided this, so we could respond to this criticism. Yeah, I think in general, as in the moment you start using publicly available data, you encounter a lot of problems in terms of comparing across data sets. This was a long answer.
No, it was interesting. Thanks.
More questions? We can take one more eventually. If not, I will stop recording. And thank you so much, Martina, for joining us.
Thank you for having me.